So in this lab, we are going to, or in this lecture, we're going to introduce the RNA-seq dataset that we'll be using on and off again for labs for the rest of this semester. And these are data from what is called the IL-10 double knockout mouse model. So if you're used to thinking about reading these symbols of gene names for mice, IL-10 is the name of a gene. IL-10 is an anti-inflammatory cytokine. This is its structure. This is a protein that inhibits inflammation in the gut. So this double negative sign here means that these mice are genetically engineered so that this gene is missing in the mice. And since this is an anti-inflammatory cytokine, it means that these mice are more prone to inflammation. This IL-10 double knockout model is a very well established and very well studied animal for looking at disease phenotypes associated with inappropriate inflammation. So the data set that we're going to be using, again on and off again over the course of this semester, starts with mice that were raised in what is called a no biotic facility. So this is a sterile facility. So these mice are raised without any microbes at all. They've never been exposed to microbes. And then the mice are inoculated with a specific strain of pathogenic E. coli. And we'll talk about why we're interested in this E. coli in a little bit. And then at 2 days, 12 weeks and 18 weeks, there are fecal samples collected from the mice. Then there is RNA-seq performed on the fecal samples. So the RNA-seq produces a large number of reads from these mice at 2 weeks, at 2 days, 12 weeks, and 18 weeks. There's a large number of these RNA-seq reads. Each read is from a bioinformatics pipeline assigned a gene identity by mapping to known E. coli genomes. So the resulting spreadsheet looks something like this. So we have our samples at day 2, week 12, and week 18. Somewhat confusingly, these week 18 samples start with W20, but they are in fact week 18. So these samples at day 2, week 12, and week 20. Each one of these columns represents the sequences from a different mouse. And this number here represents the number of times that each sequence was observed for each gene in that mouse. So this is some gene here, right? And so we would read this as saying, in this 12-week time point from this mouse, in the fecal sample, there were this many genes that were expressed by the E. coli that were mapped to a known gene. And this is the gene ID. Now, if you care about what the gene is, in a second table, so these files are available at this GitHub address here. There's a second table that's included in that distribution that would tell you, okay, well, this gene is its RefSeq ID, and there's some small annotation about what that gene is. Now, normally you would care a lot about what these genes are because if you're writing a paper in biology, you care about what the biology of each particular gene is. But in this class, we're really only considering these examples of statistical models. So we might assign a p-value to a null hypothesis that this gene is different between day 2 and week 12, but we're not going to be getting into very much the biology of what these genes are. If you're interested in the biology of what these genes are and more about the biology of these systems, the dataset that I'm describing to you today and that we're going to be using is well described in these papers from the lab of Christian Joban, who is my collaborator on these projects. Christian was at the University of North Carolina at Chapel Hill when this work was done, but he's now currently at the University of Florida at Gainesville. And I'll point you to these really interesting papers. There's lots of really interesting biology and things to think about in terms of the way that the microbiome can impact cancer in these manuscripts. So the data set that you have is described in the second paper. It's figure 3 here. This is what's called a PCOA ordination. So we're looking at now, remember there was two days, 12 weeks, and 18 weeks. And we're going to learn a lot about these PCOA ordinations later on in the semester. This is an eigenvalue decomposition. But for now, we can just say that if 2 samples are close to each other, it means that across all of the taxa, those samples were more closely related. And if 2 samples are further away, they're less well related. And what you can see is that time point is completely determining, or nearly completely determining, the structure of the transcriptome of these E. coli microbes, right? So the 2-week samples cluster completely separately from the 12-week, cluster completely separately from the 18-week samples. Now you might ask, why are we putting E. coli into mice? What is the question that we're trying to address? And really, this reflects this idea that the gut microbiome can impact human disease. And so there's, I think, a central hypothesis of a lot of the work that's been done in the gut microbiota. And the central hypothesis says that healthy people basically live in harmony with their gut microbiota. So the immune system is well tuned to the commensals that are there. But in the case of disease, something goes wrong between the balance between the host and the microbes. So maybe you have an inappropriate pathogen that comes in. Maybe you have a shift in the microbial community composition for some reason. Or maybe you have a change in host phenotype. For example, you've lost expression of this IL-10 gene. Remember, the IL-10 gene inhibits inflammation. So something goes wrong in dysbiosis, upsetting the balance between pathogens and host, and you have inflammation. And once you have inflammation, you're of course well on your way to a whole suite of diseases that are associated with inflammation, including IBD, inflammatory bowel disease, and you're at much higher risk for colorectal cancer. And in particular, this E. coli that we have the transcriptome from in these count tables that we're looking at are particularly interesting because E. coli is a pathogen that can often promote cancer. And one of the first experiments that really demonstrated this directly was performed again by Christian Joban's lab in 2012, and this is a really interesting paper. So this is looking at inflammation score in mice that are wild type. So the wild type mice are not missing the IL-10 gene, right? So they don't have inappropriate inflammation. And when you just put them in what's called a specific pathogen-free environment, so you're looking just for microbes, you put them in the animal facility. The animal facility is maintained, so there are no pathogens in the animal facility, but the animal facility at UNC Chapel Hill is not controlled. They are not necessarily strictly controlling the microbes that these animals are exposed to, but because the animals have the IL-10 gene, they don't have high inflammation. So when you look at their gut epithelial cells, the columnar cells appear normal, and you don't have inappropriate inflammation. When the mice are missing the IL-10 gene, you can put in different microbes. So this is E. faecalis and E. coli. And these are very different microbes. E. faecalis is a normal gut microbial commensal. It's kind of supposed to be there. But either E. faecalis or E. coli can drive these mice to inflammation if they're missing the IL-10 gene. But only the E. coli can drive the mice to cancer. So you get inflammation with either one of these bugs, but there's something about the E. coli genome that can drive the mice to cancer. Now, we actually know a great deal about what it is in the E. coli genome that is driving the mice to cancer. There's a region of the genome called the PKS island, and there's now overwhelming evidence that this, both in mice and it's increasingly looking like in people as well, that this particular region of the genome drives the mice to cancer. But in the data set that we have, in fact, this is the PKS genome that we were that I was just describing to you. But in the data set that we have, we could ask questions about what happens at these different time points. So in general, 2 days, mice at 2 days look like this. That's before inflammation happens. So remember, this is 2 days after the mice have been inoculated with E. coli. So the mice start sterile, they're inoculated with E. coli. After only 2 days, there's not inflammation. After 12 weeks, there's a fair amount of inflammation. And after 18 weeks exposed to these E. coli, the inflammation progresses to cancer. So this transcription data set that we have, looking at 2 days, 12 weeks, and 18 weeks, can really be thought of as a measure of disease. There's very little disease. There's no inflammation in the mice at time point 2. At time point 12, this has progressed to inflammation. And at time point 18, this has progressed to full-on cancer in the mice. And the question that we can ask with this data set is which genes that are expressed in E. coli are associated with this transition going from no disease to full-on cancer in these mice. So this is an interesting data set to look at. We're only really going to scratch the surface because we're not really going to go into the biology, but I hope this sort of motivates why this data set is biologically interesting. Now, I've made a number of adjustments. This count table is actually missing some information because if you actually look at the gene expression in the from the E. coli, remember that I said that each one of these genes is mapped to a different, each one of these sequence reads is mapped to a different E. coli gene. So we have a log 10 scale of sequence genes from 2 samples. Actually, this is from a paired end sample. We can this is from 1 sample from the Illumina run, looking at 2 different regions of the same gene, right? And you can see it doesn't actually matter which pair you look at, whether you look at the 5' end or the 3' end of our paired reads, you get the same answer. But the most highly expressed reads are actually not genes; they're ribosomal RNA. And ribosomal RNA genes are not transcribed from ribosomal RNA. This is the 16S ribosomal RNA and 23 ribosomal RNA. So genes are not expressed from these. They're structural RNA that's used to make RNA. It used to make ribosomes, which makes proteins, but these are nonetheless the most highly abundant signal in the data set, and close behind them is simply reads that didn't map to anything. So there's lots of different ways that we might have dealt with these data, and in a real paper we would think about ways to deal with these data, but in this case we simply removed them from the data set. So the count table that you have simply removes these potentially troublesome 16S and 23S ribosomal RNA. So we're just going to sort of ignore what to do about the fact that this is such a dominant part of the data set in this case. And again, this is if you're looking at eukaryotic RNA, this isn't really a problem. Eukaryotic RNA because you can use a poly A tail from human or mouse cells or from another eukaryote, so that you can select against these during the actual RNA transcription, isolation, and sequencing in a way that you can't with bacteria. But again, in this case, although I'm in the spirit of being complete, I'm telling you that these sequences are removed from the data set, but that in fact they're not in the table that you have. So the table that you have is simply going to look like this. And the very next topic that we'll talk about is how do you begin to address normalizing a table such as this one.